Hello. Boris, my man, how are you? Unmute.
Hello, Dr. Bushan.
Hey, how are you? Good to see you, man.
Good to see you too.
Yeah, let me increase the volume of my...
Yeah, I tried to connect, but it was saying the meeting hasn't started yet.
Yeah, I think there are a lot of participants already there, so...
Yeah.
So how are things?
Things are good. Students were doing examinations for the last few weeks, so we're busy with marking, with grading, and those things. But
Welcome to the real world, right?
Yeah, I can remember how in your class I used to grade only one class for the whole year, the entire year. Now this time it's like several classes every semester. So
Yeah, I'm teaching 3 courses this semester, believe it or not.
oh wow, that's unusual.
You didn't expect that from me, right?
Yeah.
Yeah, I'm doing the epidemiology, the One Health 340, and the case study is 456. So
the One Health is a new course, right?
it's a new course, yeah. It's all in the
Yeah.
package of the One Health minor.
Yeah. Are you still planning on sending students like after the coronavirus thing?
Yes, we are going to make another module. So instead of doing 456, they can do an externship in another lab or another place in another country. So that's what we are looking at, yeah.
Okay.
And one of the places we are really looking at is Mumbai Veterinary College.
Yeah.
And in your place, yeah, these 2 places we are really looking at placing them.
Okay.
Anyway,
how is the
I didn't. You were saying something?
I was saying, how is the situation with COVID now at Penn State?
It's getting better, man. We are all getting vaccinated like crazy here. So I just got both my shots. I got both of my vaccinations done. Yeah, me and Lata and everybody in our lab is now vaccinated.
Oh, that's great.
Yep.
Yeah.
So I just pulled off the isolates from your poultry genome study.
Okay.
And I'm probably going to be looking at biofilm formations in it.
Okay.
And then at some point, I'd like you to help me pull off some, the genes associated with biofilm formation there.
Oh, so you mean the isolates from the
poultry?
The poultry.
The 56 isolates where you and Rashmi were working together to get it sequenced.
Rashmi or okay, I see. Yes. No, yeah.
Yeah. So that data is coming out really well, and that's the next big push. We need to get it out in the market. Yeah.
Yeah, that's
some
good.
awesome data. The data is just fantastic.
Oh, great. That's good.
Yeah. And I'll ask Ling to share those data sets with you, because ultimately we would like to close some of the genomes there.
Yeah. So did Rashmi sequence all of them?
Yes. Yeah, Rashmi and Ling, of course. Yeah.
The next seq, right?
Yeah, that next sec, yeah.
Maybe we'll need to throw in a couple of samples on Meera. And,
yeah, that's what Ling and I have been talking about. So
and 2 flow cells should be fine with 60, and we don't need to sequence all of them, right? We don't need to cross
no. As long as we get the CGMLST codes, if they all align, then we can take representatives of the CGMLST. So that's the next step we need to do is identify the CGMLST and then say, okay, we found so many of them. Let's select a representative of each of them and close the genomes.
Yeah, Ling told me about that, about the CGMLST with the stuff. And the databases that we've been using, the tools we've been using don't have stuff or rests in them. So I told him that I can write a kind of a script that can do the same thing, because the same thing we've been doing with Salmonella. We can
that's what you did for Salmonella, right?
Yeah.
Hi, Meera. How are you? I see that you sent me an email at 2:35 in the morning. What were you doing so early?
My work timings are a little bit shifted based on her sleep time. So I had to wait until she sleeps to get some work.
Okay.
Hi, Meera.
Hi. Can all others hear us?
I'm sure everybody is hearing us. It is before we start. It's okay. I'd like others to join in too in the conversation. So
Right. I had some issues first with the speaker, so I think I figured it out. That's why I had to log out and log in back.
Okay. Yeah, it was pretty simple for me. I just clicked on it and I got right into they are running a WebEx-based platform. Yeah. So how is the COVID-19 sequencing going on, Meera? Any idea in your lab?
Yeah, some interesting stuff's coming up. So we are sequencing both community surveillance, trying to, and we have few of them, and we also are doing some wastewater screening. So we are seeing some variants in it. So that's interesting. So we want to do more sequencing and see, are we really seeing any immune escape variants in that one? And we also want to see the options to sequence the surveillance program samples, which we are not doing it at now. This is just a surveillance now, not a research project as of now. So we want to figure out the feasibility to try doing that.
So Ling was telling me that you're going to move into first doing, putting it on hey, Umesh, how are you, man? You're muted.
Thank you, Bhushan.
Hey, good to see you.
Good to see you. I knew that I was trying to get in touch and connected. Yes, since yesterday I was trying. But,
yeah.
Let me. Okay, carry on. Sorry, everybody.
No. We were chatting. I don't know. It's probably another 10 minutes we will start. So I rarely get to see Morris. I rarely get to see Meera, although I'm just 200 feet away in the lab. This has been the best chance for us to keep talking. Yeah.
Yes.
How are you doing, Kumesh? How are you?
I'm fine, thank you, Bhushan. How's everything at your end?
Doing good. Hey, I'm coming on 14th May.
14th May. Okay, perfect. That's fine.
Yeah.
How's everybody else in the group?
Doing good. Everybody's getting vaccinated left and right. So hopefully we think that's the cure for everything, but not.
At least we can try getting ourselves vaccinated and try to be as strong as we can. Nothing else. Did you take your vaccine, Bhushan?
Yeah, I finished my both doses. Yeah.
Okay, I did my first and I'm waiting for the second. Now the interval is gone beyond 6 to 8. the new guidelines suggest to probably another 2 weeks to wait more for the second shot.
Yep. Hey, Morris and Meera, the person I'm talking to is Dr. Umesh Karkare. He's my friend, my mentor, my advisor in Mumbai. You know, we go back almost 30 years. He's one of the very well-known small animal practitioners, and he has come to the US several times. He has spent time in our lab too. So Umesh is one of what we call as the innovators and early
really
adapters in doing things. Yeah.
Nice to meet you, Doctor.
When I was reading the today's program, and I said, probably I'm going to meet Bhushan. It's a proteins. Proteins. Come on. I was very sure actually. Yesterday I tried to log in, but somehow I couldn't get through. It was very difficult. I tried at 3 o'clock lecture, 4 o'clock. Before yesterday, 3 o'clock lecture, I couldn't get it. 4 o'clock. Yesterday I missed, I think it was at 5 o'clock, if I'm not wrong. I think every day the time is different. So you have to go back, check, and then find out a time and then log in again.
Morris, you're what, five hours ahead of us?
6 hours now.
6 hours now, okay.
It was 7 hours before March. Now you came back forward, so we are only 6 hours.
So is Wasker going to be doing, or is Zende who is leading the process here?
Bhushan Zende is there. Probably he will be.
I think so, yeah, because I had a quick chat with him 2 days ago, and he said Wasker, myself, and another person will be the three of us who will be doing this. So just, you know, since we are approaching, we got another 7 minutes left. I was wondering if I should wait till 8:30, see if somebody from their part comes up and starts the process. Yeah.
Okay. Bhushan, I'll join you in 5 minutes now, okay?
Okay, good. Sure. Take your time. Take your time. Yeah. Yeah. Hello. Is that Wasker? I'm not able to hear you. Wasker.
Hello. Good evening,
Morning,
everyone.
Zende.
Sir, namaskar. Good evening.
Good evening. How are you doing, man? It's been a while. Yeah.
Thank you, sir. Yeah. Dr. Morris.
Hello.
Yeah. Good evening.
Good evening.
Yeah. So,
I think it's morning for some.
yeah. Sir, we'll wait for another 2-3 minutes
Okay.
because people they are joining.
Okay.
Okay,
No
sir.
worries.
Yeah,
Yeah,
yeah.
For us, it's 8:30 in the morning for me and Meera.
Yeah.
So,
Yeah. Yeah. Meera is also joined. Meera, good evening. How are you
Evening.
doing? Yeah.
Good. Thank you. How are you?
Fine, thank you. Fine, thank you. Sir, Dr. Patukar, sir, our Honorable Vice Chancellor, I think he is having meetings. Still is in the meeting. Just he is
okay.
called me. So he asked me to just go ahead. In between, I will join.
Okay, that's fine. Yeah,
Okay.
yeah,
Okay.
it's
Yeah. Yes.
So let me know when you'd like to start off, and then I can introduce Morris to begin with.
Yes, sir. Yes, sir.
And then when
Another
Morris
2
is
minutes.
done,
Sir. 2 minutes. Only
take
2
your-
minutes. Wait.
yeah,
Yeah.
just let me know when you'd like me to start. Yeah.
Okay, sir. Okay.
So is your BSL-3 lab completed now, or what is the phase? You said you indicated your lab is.
Sir, already we are done with clean room. That biosafety
Okay.
room is now ready. It is commissioned, and also the virtual classroom is also ready. But
Fantastic.
yeah. And I got most of the equipments like real-time PCR, Quantum 5.
Okay.
Then VTech machine also I purchased. VTech 2.
To do your multi species identification.
Yeah, species identification. Yeah.
Okay.
Yeah, and about the other equipments like sir MDS that digester for the ICMSF and also for the ES also I have purchased that. LCMSMS I think sir LCMSM and ICPMS I think they will land in India by tomorrow evening or day after tomorrow.
Okay.
Yeah, and rest of the other small equipments are there, so that also we have purchased. So
Okay.
by
So
so by
another.
30%, yeah, by 30% March, I think the whole equipments will be there and it will be established and little bit work will be. And but sir, we are also trying to develop certain training programs further in the month of April and May.
Okay.
So
Let me know if there's anything that we can help you out. Yeah.
yeah. So like detection of food pathogens that we have to have in the month of April we are planning. I think
Okay.
this,
So the person is right here with us. Meera is our
yeah,
food safety
yeah,
expert.
yes,
Yeah. So
yeah.
Meera will be, and she has been working on it for the last 10 years. Just
Okay,
microbial pathogenesis and diagnostics she'll be a
yeah,
perfect fit. Yeah.
yes, sir. So definitely,
And
and also, sir, you advised me to that epidemiological approach for the veterinarians, including all academicians, then students, and so the private practitioners. So that
right.
also, and also the veterinarians working in the field. So that also we have to plan for about 1 week program or something
Yeah,
like that.
we can do a field epidemiology based program course.
Yeah,
It's called as
yeah.
field epidemiology, where people in the field can use epidemiologic methods in
Yeah.
order to understand their settings. Yeah.
Yes, sir. Yes. Dr.
We
Karkar,
can
sir,
do that.
good evening, sir. Dr. Karkar, sir, I think you're muted, sir. Just unmute yourself, sir.
Hello everyone.
Yeah. Good evening, sir. Sir, yeah. He is just adjusting.
Mhm.
Hello everyone. Ravi.
Hi, Oscar. How are you?
Hello. How are you, sir?
Hello.
Doing good. Yeah.
Yeah, fine.
You've gone and hidden yourself in Satara. Why?
yeah,
Of all the places, leaving Mumbai and going to Satara, why?
yeah,
Why
it's
not?
a little offbeat place now.
Yeah.
Yeah, Bhushan, how are you doing?
I'm doing good. Yeah.
Yeah, that's great, sir. That's great.
Yeah.
Ravi, it's 6 o'clock.
Yeah, it is 6 o'clock. Okay,
Yeah.
sir, now we can start. I think now there are 64 participants. I think they are just in between, they will join our training
Okay.
program. So I welcome you all, sir, on third day of this 1 week international e-training program on introduction to bioinformatics, microbiome. So today is the third day, and we have today the great personalities like my teacher, my philosopher, Dr. Jairao sir. He will be acting today as in chairman and moderator for this today's training program. So I welcome two speakers also here, Ms. Arpita and hello, am I audible?
Yeah, we can hear you. Yeah.
And Dr. Morris. So I welcome you all, and Dr. Karkar sir is also there. I welcome you all on behalf of my NHP team and our MAPSU authorities for this training program. Our Honorable Vice Chancellor and Director of Research, Dr. Kurkure sir, and Dean Faculty of Veterinary Science, Dr. Sonkur sir, they are busy in, because sir, this is a March end, and the people they are just involved in committing what are the expenditures they have to make up to the 31st March of this current financial year. So these people, university authority, they are busy in that. So they assured me, so you can go and you can start the programs in between. So definitely they will join to hear our learned speakers today. So, so in this training program, we have two eminent speakers today. Dr. Morris, he will speak on the shotgun metagenome sequencing concept and approaches, taxonomic and functional analysis. And Dr. Meera Nair, she will also going to speak on microbial community analysis, concept and approaches, clustering and diversity analysis. So I welcome speakers and all our participants across the country and from the abroad also for attending this training program. And I hand over the mic to my beloved teacher, Dr. Jairao sir, please.
Thank you, Dr. Zende. It's truly a pleasure to come back to Bombay Veterinary College, and more so in a role as a collaborator and working as a partner with MAFSU, as we have just recently signed a MOU with you in order to help with some of the programs that we would like to foster with you over the next couple of years. So with that, thank you so much for including us, especially the Penn State Animal Diagnostic Lab, to be a part of your training program. And we'll try and do our best to share with you that what we have been doing here and what Meera and Morris will share with you. To begin with, let me start off with Morris. Morris is going to be our first speaker, and it's my distinct pleasure to introduce Morris. I've known Morris over the last 6 years as my student, my friend, and my colleague. So it's truly been a pleasure to have Morris as a part of Penn State. So Morris is a native of Rwanda. He obtained his Doctor of Veterinary Medicine from the School of Veterinary Medicine in Dakar, Senegal in 2008. And in 2010, he obtained a master's in biotoxicology. And then he worked in University of Rwanda from 2009 to 2015 as assistant lecturer. And in 2015, he was awarded the very prestigious USAID Board Fellowship to do his doctoral studies at Penn State. Little did he know at that point in time he was getting into too much trouble with Bhushan. But it has been it was wonderful 5 years of working experience with Morris. So he joined my lab in 2015, and in 2019 he obtained his PhD in pathobiology. And I've had the good fortune. I've had a good run so far. I've mentored 21 PhD students and 12 masters. And all I can say is Morris is one of my best students I've worked with. and he's a brilliant scientist, and I'm very proud of his accomplishments. So today he's going to be talking about application of genomics and bioinformatics to metagenomic-based approaches. And today is going to be the lecture part of it. And I believe tomorrow he's going to share with you some of the tools that he uses in order to accomplish metagenomic analysis. With that, I will stop here and ask Morris to start his presentation.
Thank you, Dr. Bhushan. Thank you for the kind introduction and the kind words from you. And I think the time I spent with you in your lab, actually, I would say that pushed me towards what I am now in terms of career and professional. I'm thanking you also to have connected me with this wonderful group and given me this opportunity to share with the group a few of the things that we have been doing, not only for animal diagnostics, but also trying to investigate pathogens in the environment and in animals. I'm going to share my screen, and I hope you can hear me well. If not, then you will let me know, and I will increase the volume of my speaker. Okay, can you see my screen now?
Yeah.
Or?
We can see your slide, Morris. Yep.
Okay. Okay, so like Dr. Jaro mentioned, we're going to take a few minutes, like 60 minutes, talking about the theory part of the shotgun metagenomic sequence, the metagenome sequencing, especially the two approaches of this technology. We'll talk about taxonomic analysis and functional analysis. And today we will just look at the theory aspect of it. And tomorrow we will do a demonstration of some of the tools which are used to do the analysis that we will talk about today. I want to thank all of you who have sent the responses to the survey. This was really very helpful for me to have an idea of how I should organize these sites. The reason is because there are so many things that we can talk about in metagenomics. To put all those things into just 1 hour requires just going over a few things, or maybe the general aspects, to make sure that we understand the general principles behind metagenomics, but also the general idea, the general tools that will help us to understand the analysis or the output of those analysis tools. So we will start with a couple of terminologies, so a few terms that we need to understand, to have a common understanding, because there are a lot of misconceptions about metagenomics. We want to clear those right away. Then we will have an overview, maybe 1 or 2 slides about how the metagenomic projects are designed. A couple of words on sampling considerations when we are doing a metagenomic project. Then we will have an overview of some of the analysis pipelines for both taxonomic analysis and functional analysis, and see how these differ in application. So one of the term that usually causes confusion is the microbiome, especially how does it differ from the microbiota. These days we are every day bombarded by terms ending in omic. Everything you can put in omic and then it becomes a new term. This is because we are using computers to solve problems, and every problem we can solve with the computer using the genome, we want to put it omic on it. So the microbiome can be seen as an omic word, just like any other word like microbiomic, transcriptomic, and others. And in that case, biome will be considered as a collection of the genomes in an environment. But some people believe, and myself included, that microbiome is rather a biome world, meaning the microbiomes. This refers to the collection of the microbes, the microorganisms in the environment. And in that case, the genomics, the microbiome, in terms of metagenomic, will be a tool to use genomes to understand the microbiota or the microorganisms present in an environment. So metagenomics, meta, you probably already know, means beyond. So metagenomics comes in to emphasize the fact that with metagenomics, we break the barrier of a single genome. So bioinformatics have been dealing with a single genome, single organism genome. You sequence a genome, and then you try to find out what are the components, or what are the things you can learn about one genome. But in this case, we want to learn about all the genomes in the environment, because most of the functions of the bacteria or other microorganisms will rather be in concert with other organisms instead of just individual activities. So again, this is another word which can cause, because I've heard people talking about microbiome and then metagenomics as if they are two different things. Rather, these are two different approaches of one thing. So metagenomics can be targeted, which means we use a marker gene. I think this is something that we'll hear in the next talk. And non-targeted mean shotgun metagenomics. We will talk about this in more details in a few slides. So those two approaches are illustrated here. So with a marker gene like 16S or ITS in RK, we can see that a single fragment of the genome is sequenced, and because that single fragment carries information about the diversity of microorganisms, we can use it to make a taxonomy or taxonomic classification, as we will see later. And also, since we know which organisms are probably present in an environment, we can use that information to predict the function or the functional potential of the microorganisms in that environment. On the other hand, the shotgun metagenomics is where you have all the microorganisms in the environment, then you sequence them indiscriminately. You don't just want to sequence a part of the genomes, you sequence everything that's there. In that case, you can also do the taxonomy, but of course, as we'll see, or you can understand from here, this will have a limitation in terms of how deep we can go in terms of richness of our sample. But it has an extra advantage that we can get information about the strains that we may not probably get just using a marker gene. And an additional and most important advantage of shotgun metagenomics as opposed to marker-based metagenomics is the determination, not just the prediction, because you can directly identify, you can directly classify the functional potential of the organisms in the environment just using metagenomic data. And for this next slides, we will just focus on shotgun metagenomics and its applications. So some of the advantages I was mentioning is that using marker genes requires primers and amplification. And when you do that, if the primers you use are not appropriate for some microorganisms, those microorganisms will be missed because you won't be able to amplify them. But with metagenomics, it assumes that you sequence all the DNA you have in the environment, or all the RNA if you're using metatranscriptomics. So, and you can do all of them in a single run. And most importantly, as I said, you can do a functional analysis, which is not possible with marker genes. And so this is one study that was looking at the microorganisms in pigs. You can see that you have the potential not only to analyze what is already known, like eukaryote, bacteria, viruses at the same time, but also there is a potential to discover new things, because as you can see in most of the studies that have been published so far, there's always this component of not assigned sequences, which may potentially carry new pathogens that may be of interest tomorrow. Now, so we'll try to use this flow of events from the experimental design to the data analysis. Any metagenomic project will have all these steps, but some of them may not be required depending on which type of analysis we want to do. So we will start with the experimental design, and I think these two, experimental design and sampling, are the two most important parts, two most important steps of this process. The reason is, if the experimental design, the experiment is not well designed, the data that will come out of the experiment will be flawed and may not be used to get you to the goal you set for the project. Same thing, the experimental design might be good, but the sampling is wrong. That may cause a problem of representativeness. So you have the DNA which does not represent the DNA environment, and that will be, of course, to give you wrong results and interpretations, and so forth. So, and then there are steps here like assembly. You can see that there is a dotted line. These dotted lines mean steps that we can skip. So we can skip sample fractionation, for example, depending on the amount of sample we have or the design of our experiment. We'll see later in a few slides later, we'll see that the assembly is not always required. The assembly is required only when we're using a technique called binning when doing the analysis, taxonomic and functional analysis. If we are not interested in binning, we may not be interested also in assembly. We can go right from the DNA reads, the DNA sequences, to the annotation of the function, and that will be it. So again, today we're going to focus more on the analysis part, but it's important to understand, first of all, the design because it has lots of implications on the analysis. So when designing a metagenomics project, it's very important to think about control groups. So these control groups you will see in most of those projects, that will always be normal versus disease, normal versus cancer. It's because we want to make sure that what we see is not just due to errors or contamination and others. And also there are several considerations which are dependent on the goals that you want to achieve. But the control and also variables which might confound the control comparisons are some of the important aspects to consider. Again, for the sampling part, we need to make sure that what we have matches what we have in terms of a sample matches the goal that we set ourselves before we start the metagenomic project. So one of the things is the sample collection and the preservation, because in some of the metagenomics, you want to, for example, look at the differences in a sample over a period of time. So you may want to see how something evolves after like 1 year or 2 years. So in that case, you may want to take a sample and then take another sample after 6 months, take another sample after 1 year, and then sequence all of them at once. So you understand that there's going to be a very rigorous storage conditions that you have to respect. So sample correction, then sample storage, very key to make sure that the sample, the DNA in our sample represents what was there at the time of sample collection. And of course, the negative controls will be super important as well. And another thing that, so when we do metagenomics, we need to make sure that the sample, the DNA extraction methods we use are appropriate for metagenomics, because sometimes we do DNA extractions for other applications, maybe for single genome sequencing, if you have like pure bacteria, pure isolates of bacteria. The same DNA extraction kits you use may not be the same you want to use for metagenomics, because a metagenomic is a kind of complex sample, and complex samples require specific kits or specific methods for extraction, which have been optimized for that kind of sample. Then one of the things that we come across is you may have maybe let's say a sequencer in your lab, and then say, okay, so I have a sequencer, why can't I do just a metagenomic project? So there are a lot of things you need to think about before you just start collecting samples or launch a metagenomic project. And one of them is how much data you will need, because as we will see later, that will depend on what kind of hardware do I need to get to that amount of sequence. So there's this paper in Scientific Reports that determines that you can use to determine how much data you need based on the target you want or the goal you want to achieve. So from that paper, so there are formula. I think there's a formula that you can use depending on your target to know exactly how much data you need. And what they based on here is that for us to be able to assemble a genome in a complex sample, in a metagenomic sample, that genome has to have at least a coverage of 20x. So based on that principle, and this was not just something that came out of the blue, this is something that came out of an experimental approach. So now in that paper, they used a target. They set a target of 1% of the relative abundance. That means that if you have a sample in an environment, or maybe you took a sample from an animal and you want to be able to sequence all the things which are at least 100% in that sample. So you're not interested, for example, in organisms with abundance which is less than 1%. So if you need to sequence to be able to analyze, to get the genomes from organisms of 1% in terms of relative abundance, you will need 7 gigabases of data, not gigabytes, gigabases of data. So this includes if you are sequencing with a paired end, if you sequence with Illumina, where each read will be sequenced from both ends, then you will need 3.5 for each read, and then the two of them will make up 7 gigabases. So as an example here, let's say you want to see what's going on with some kind of disease. You get a pig or maybe a group of pigs and then you have a normal pigs as controls. You want to have technical replicates for each of them, and of course you have a negative control. Each one of these technical replicates will have to be 7 gigabases if you want to be able to detect those low abundance organisms, and the total will be 35 gigabases for this simple experiment. In the real time, you will not just do this, because in real time you will want to do also biological replicates, which means that you will probably use different animals or maybe the same animals in different conditions with the same disease. And those biological replicates will mean that everything you have here will be multiplied by the number of biological replicates, which may increase tremendously the amount of data you want to achieve. So that is just a few things. Now there are a couple of other things that you can think about is the spatial strategies, because I would say that the classical strategy for metagenomics, just you take the genome, the DNA, you just do the library and then sequence. There are sometimes, if you look at the same figure we presented before, you're going to see that the viruses are only 0.09, so just below 1%. So if we use the same strategy, we may miss a lot of information on organisms with low abundance, like viruses. That's why there are techniques that amplify some organisms, for example, the random prime amplification, which can amplify randomly, which can not targeting a particular virus, but we can amplify all the DNA you have in a sample. Another strategy, which I think is currently which is being done in Jairo's lab at Penn State, is what's called CISPA and GS. It's also a kind of independent. It's called the sequence independent single prime amplification. It's in the same kind of approach, where after you have your sample, you selectively remove the host genome before you sequence, because as we will see later, most of this step of removing host genomes after you've sequenced. But of course, if you do that automatically, that will not increase the amount of the data you have from other microorganisms. So by removing the host genome and the environmental genomes, the non-wanted genome, that will enrich the nucleic acids from the microorganisms, which will increase the ability for us to detect even those that have low representations. And that, of course, will most likely tell us what are the things that we didn't know exist that we can detect bioinformatically. So after those considerations, after you know how much of the data you need because of the target you've set yourselves, this will now bring us to which instrument can we use. So if you have in your lab just a MiniSeq or a MySeq, you cannot even do that simple experiment. Of course, there are things you can do. You can, for example, have a library and sequence it several times, but that will be, first of all, it will be more expensive than when you use just one sequencing run. But also, it may give us false results which are difficult to analyze, because you need to do things like so things like what is it, harmonization, and that's not something we want. So if you sequencing run, you've calculated maybe you need 70 gigabases, a next seek will be enough. And in fact, the next seek is enough for many of the medium level projects of metagenomic. There are other instruments like HiSeq, which are kind of industrial scale, and NovaSeq, which is not here, which can be used. Also, one other thing that currently is being used is the long, what we call third generation sequencers, things like Oxford nanopore sequencers, MinION, GridION, and other things. So depending on the maximum output expected from those machines, we can sequence our metagenomic library with some of them, but probably not with a MinION, although a MinION can do well with just a single sample, but not when you want to do a comparison between samples. Of course, we need to make sure that we know what exactly we want, because if we want to have a high accuracy in our taxonomic analysis, then this may not be accurate. But if we just want to detect the presence of a particular organisms, then these third generation sequences with long read potential will be enough, because they provide us enough data to detect things, but not maybe comparison based on mutations. Okay, so now we've sequenced, we're now moving away from the bench to the analysis, to the computers. So after we have sequenced, and of course, as you can see here, I didn't talk much about the library, because that would be another part of the training. So a FASTQ file is mostly what you will get from the sequencers. I believe that you may have heard this part in other parts of the training of this workshop, but I want us to know what is a FASTQ file. The FASTQ file, because we will see later that some of the tools that we use in bioinformatics will allow us to use FASTQ files. FASTQ files mean the raw files that we get from a sequencing machine. So a FASTQ file will have sequences or reads, as we call them, which are represented by 4 lines. The first line is the ID of the read, and then followed by the sequence itself, followed by plus sign. This plus sign will always be just a plus sign, nothing else, just the format. And then the Q score. This is super important. We'll talk about this again in the next slide. So the Q score is very important for us to know whether we can go ahead and use that read, or if we can just get rid of it. So if you want to count the number of reads in a file, you just take the number of all the reads in that file, then divide that by 4, because each read will be presented by 4 lines. So now the analysis metagenomics pipelines now, so let's talk about those quickly. I know I have only 1 hour to get done with this, but I'll try my best to go over all the slides I have. So the first thing that we need to do when we want to do the metagenomic analysis, we have the data, we have our data, we need to do the data exploration, we'll see what that means. Then the pre-processing, this is an important part, which is part of the quality assurance, to make sure that we have the good quality data, because the quality of the data will also imply the quality of the analysis and the quality of the interpretations. Then we do the taxonomic analysis and functional analysis, so we'll see what that means later. So the data exploration is, I think some people like to automate stuff, to automate things, but I don't think it's always a good idea. I think this part of data exploration should always be done manually because this will require a judgment of a person doing the analysis. And one of the things that we can do is to make sure that we have sequenced all the samples. You may have like a number of samples, and for some reason, one of the samples didn't get sequenced. We need to make sure of those, and then look at the file sizes and see if the size of these samples is what you expected, depending on what you've put it into the machine. Things like demultiplexing, making sure that the indexes you used have been demultiplexed correctly. The next step, again, in the pre-processing will be the quality control. The quality control, the first thing with the quality control will be the quality visualization. So a tool like FastQC, which is I think the only one actually I use to look at the quality of the FastQ files. Some people may know, probably others, but this works fine with me, especially the per base quality scores here. If you look at this slide in the FastQ files, we have good data because we have a lot of data in the green zone, but all the data, all the reads here, all the segments of all the reads in the red zone are the reads where the average quality is below 20, and we don't want to do analysis with reads with a quality that is in twos. Usually when you have like the newest sequencing machines, if you have good quality DNA, most of the reads will be in the green, a few of them in the yellow. But if you have data like this with all these reads in the red, you want to trim them to remove all those that are below a given quality score. So some of the tools which you can use to do that, Trigonometric and QTADAPT, I almost exclusively use Trigonometric. The reason why I do that is because it works for me, and when something works, I don't want to do other things because there's nothing that tells me QTADAPT will do better than Trigonometric. And also, Trigonometric will also remove, I think QTADAPT is QTADAPT as well. The QTADAPT will also remove the adapters, but you need to do that only if you have a red here. If you think there are adapters in your samples. Then after you've done that, the quality control or the pre-processing, and in fact, we will be doing this. So we will have the demonstration of these things tomorrow as part of the demonstration. So the taxonomic analysis is the first thing that we do in a taxonomic, in a metagenomics pipeline. Sometimes the only thing, sometimes we are interested in just what is there. And you would ask me if we are interested in what is there, why don't we just use the marker-based like 16S? It's because sometimes what is there is not known, and we want to know it, and you can't have primers for things we don't know. So if we want to do viruses and we want to find out which viruses are present in a sample, at the same time, what other organisms are together with those viruses, the marker gene will not work. So the taxonomic profiling will be used in that case because we want to know what is there, not necessarily what they're doing. So what we desire is to go from the reads. So we've sequenced, we have these short reads that come out of the next-gen machines. And we want to go from the reads to the taxonomic profile. The taxonomic profile mostly expressed in terms of relative abundance. So we have a sample, then we have a number of taxons. We want to know what is the share of each taxon in the whole sample, the entire sample. So we express this usually in terms of percentages. So now, how do we go from the reads to the taxonomic profiling, to the taxonomic classification? Well, so a couple of years ago, I had a course, I took a course on Coursera. It was good because it summarizes how metagenomics can be used to do a surveillance of antimicrobial resistance and pathogens. So I took notes of some of these slides that I was using for studying in that course, and I thought it would be good also to use them here because some of you might be inspired and going to look for that course. I'm not sure whether this course is still available on Coursera, but if anyone interested in how to use this metagenomics for antimicrobial resistance, this would be a good starting point. So there are two ways to do the taxonomic analysis. One of them is to do the classification of the reads. So it means that you look at the reads abundance, the reads number, read numbers. You don't want to do the assembly. And those classification methods are machine learning techniques of classification. If anyone is aware of the machine learning techniques, one of them is the classification, which can be supervised or unsupervised. We're not going to go into the details of that, but what I want you to know is that these classification methods are of 4 types. So there are those that use sequence similarity. As we will see much later, they're almost similar to those that use assembly. And those which use sequence composition. Sequence composition are things like GC content, the GC percentage, things like the chemers. We'll see that some of the tools use chemers. And there are those which are hybrid. They use the sequence similarity and also the sequence composition at the same time, call them hybrid methods. Then we have those that marker-based. This is a little bit confusing because this is not to be confused marker-based metagenomics. So the marker-based metagenomics, you amplify just a single segment of the genome. So the marker-based classification is that you have your gene data, then you can use several markers. Actually, most of the tools that use like thousands of markers at the same time to classify those reads. We will see a few examples of those. Now, another type of taxonomic analysis is through assembly and binning. So assembly is just the same genome assembly that is done with a single genomes, but this is applied to a multitude of genomes in the same sample. Then this followed usually with the annotation which is part of the functional analysis. So today we're going to start with what are the tools that are available for classification of reads. So the classification, which is part of the taxonomic analysis using the reads without assembly. So I was looking at the available literature on this, and then I found this paper which was published in 2015. It lists a number of tools, of software, with how many times they are cited in the literature. Again, this is on 2015, so things keep changing because these things keep updated. So for example, in 2015, this Megan was Megan 4, now it is Megan 6. And we can see that Megan is the most cited, and I was curious to know why. And it's because, as we will see, Megan is very user-friendly, because Megan has a standalone, has a graphical user interface. MGRest is the next, also it shares something with the Megan, which is the ease of use, because this is a web-based tool. So you put your data into the on the server on a computer, upload, and then you wait for some time, and you get the results. Now there are other tools like the Metaflan, which is a super tool. I like Metaflan, but of course, because it's a command line tool, it has a low level of citations, but it doesn't mean that it's a bad tool. Now it uses they use different methods. So these three uses similarity. Others will use hybrid. Others will use the marker, and so forth. Now here we have others which are less cited, but I think Kraken right now is one of those that are used commonly. And also it's important to look at what are the functional classification possibilities, because some of the tools you use don't give you the ability, the possibility to do the functional classification, the functional analysis. So the three, these are the four most common used. Those I've found on forums, and those people talk about are Megan, and then MGRest. It's a web-based, it's a web server. The problem with this kind of servers is that sometimes you open an account. For example, I think I tried yesterday to open an account. But when you do that, someone needs to look at your request and send you an email. I tried it 2 days ago, and I'm still waiting for their email. So probably it will never come, or maybe I'll have to do it again. But Megan is good. Now, Megan has two options. We'll look at them tomorrow, because Megan is one of those we're going to demonstrate tomorrow. It has a free option, a free version, and a paid version. A paid version, you have to pay like $3,000 every year, but it has more potential, especially for functional analysis, because it comes with lots of databases, functional databases that we will see later. Now, Kraken is another one. It's a command line. It's a good one, and it's of course a command line which is not easy to use. But once you have just a few skills on command line, which we'll talk about tomorrow, or maybe if we have time today, can be less intimidating. Same thing as Metaflan. And Metaflan, the good thing with these Metaflans, we'll see, it's because although it's a classification method, it goes together with another tool, that the human tool, that you can combine together and get to a full analysis, meaning taxonomic analysis and functional analysis together. Of course, I put here the links to all those tools. Some of the links are not the links to the tools. Some of the links I will put in the PowerPoint are the links to the tutorials, which obviously will point to the tool itself. Now, the second part is the assembly. The second type, the second method of taxonomic analysis is assembly followed by binning. So this will assemble the reads into contigs or scaffolds. I hope we know what scaffolds are. So scaffolds are contigs which you put together because they belong together. And one of the things that is more qualitative than quantitative, you can use it quantitatively, but it's less quantitative than the previous classification methods, because if you have a lot of reads and then you assemble them into a contig, it will be difficult. It's possible, because you will see, because you can then map again the reads back to the assembly, but that is an extra step which will reduce the quantitative ability of this technique. So some of the pros of this technique is that they group contigs together, binning, and this will have a more resolution of functional analysis. The reason is it will be easy to match taxonomic composition with the functional composition. So, for example, you find that contig number 3 belongs to, let's say, E. coli or a Salmonella. And then the same contig 3, you find a gene which is a gene for antimicrobial resistance to a particular antibiotic. Now, it will be easy to know that that gene comes from Salmonella. But if we don't do assembly, it's possible, but it will be difficult because sometimes you do taxonomic, you know that you have Salmonella, E. coli, and others, you have your diversity, and you also do a separate functional analysis. You know that there are genomes, genes for this kind of virulence, genes for this antimicrobial resistance, but you don't get to know these genes are coming from which organisms. So when you do an assembly, that gives that resolution, because you can say, okay, so since these genes are coming from the same contig that came from Salmonella, so there's a high probability that these genes I'm seeing are coming from the organism in question. And again, it reduces the risk for false positives, because if you have sequenced something, if you have assembled something into a very long contig, there is a higher chance that that thing exists, as opposed to when you have very short sequences that you blast against the database. There might be a random chance for alignment to something from which it didn't come from. So another thing is that when you do assembly, there is a chance to discover new things like viruses and other organisms that may not be, that are not in the database because of the de novo assembly. Now, the cons, these limitations for this assembly is because it's super computationally expensive. So you need a huge amount of memory, RAM, and most of the computers that we use on our desktops may not be appropriate to assemble things that are of 7 GPs of raw, and also do the bidding at the same time. So these tools are really very expensive in terms of the computational capabilities, not only the hardware, but also the software. And also they have the higher detection limit. Higher detection limit because if there's something you cannot assemble, then you will not be able to detect it. So at the beginning, we talked about 7 gigabases. We talked about higher coverage, at least 20x of coverage to be able to assemble genomes which are 1%. So here, if you have less than 20%, you may not be able to assemble those reads. And we know they come from something, but because we don't assemble them, we may be, we will miss them. So the assembly, it's just the same assembly, for other organisms. The difference is that here, instead of having one organism, we have different organisms. Each color here will represent an organism. Then the bench. The first part here is the bench. You do the library. You fragment your genomes into small fragments, which will be sequenced using the next generation sequencing machines. And then they give you all the reads. Those are the reads. So these are the fragments. And then after you sequence them, they become the reads. So you align them. You assemble them using the tools we will talk about. And what you have at the end of this are the contigs that are assembled that we use for binning. You can, some of them can be scaffolded if they have the information, especially when you use things like paired end in Illumina. It is much easier to get scaffolds, which are long contigs. which are grouped contigs, which are supposed to be contiguous. The assembling methods which are used for metagenomics have to be a little bit special. We can use some of the assemblers that we use to do the normal assembly, single genome assembly. For example, you see spades. Spades can do the assembly, but spades, they also have another meta spades. So spades, these guys, they have several tools which are adapted to a particular kind of assembly. So I think I was looking at their website yesterday, and they even have the coronavirus spades. So it's a spade which is specifically optimized for coronavirus. I don't know if that's really a way for them to be fancy. But they have spades for everything, for plasmids, spades for metagenomic as well. And one of the things that it's difficult to, these things of assembly depend on people's preferences sometimes, because they have all the metrics that you can use to choose which one is good for you. You may be interested, for example, in length of some of the contigs. In that case, you have ultra long contigs, like more than 50 kb. In that case, you see that metaspades and the spades itself will work better because they have more of the long sequences. One of the metric that people use most often is the N50, which measures, it's a measure of how long in general your contigs are, because N50 is the length that carries, that carries 50% of yours. It means that if N50 here is 800, that means 50% of your reads are higher or equal to 800. In that case, you will find that some tools, I think here, Omega will score the best. But some of the times you want to just look at the trade-off between the quality and your computational capability, because the memory that is required here for a metaspade is only 66 against 157 for just spades. So sometimes you may say, okay, now I'm going to have a drop into the quality, but that is worth it given the computational easiness of the work. Now you can also look at the time. Although like this Omega, we say it's good because it has a higher N50, but it will take almost 2 days to complete a metagenomic assembly. So these are the things you can look at to just choose. I usually do spades when I'm doing microbial sequencing. I usually use spades and metaspades. They work fine for me. But some people may like this CRC because it's a graphical user interface analysis pipeline. It has a lot of analysis tools. They also take less time. They use the lowest memory because you can use it on a normal computer, but of course the quality will drop in comparison to others. So now, once you have done your sequencing, what can you do in terms of genomics? You've done your assembly. Now you can do the quantification by doing the reverse of what we did previous. So we went from the data from the DNA to the reads. Now we can try to think about how we can do the reverse. So what you do, the first step is mapping. You can map your reads now to the individual contigs. If this contig came from the most abundant organisms, then what we call the average coverage, the coverage for that contig will be higher than the coverage of contigs which came from less abundant or less represented organisms. So this is one of the quantification you can do, which is not as easy as if you do direct quantification of reads, but at least it's a way to quantify to do the quantification, the classification, the abundance of microorganisms. And of course, here we assume that if we know the reads, which of the genome has the most reads, we can infer to the abundance of that organisms in the original sample. So the next is binning. So binning is grouping. Yes, please.
Can you wrap it up in 5 minutes, if you don't mind?
Yes, I can do that.
Yeah. Take your time, but yeah. Thank you.
Yeah, I think 5 minutes will be enough. Yeah.
Okay.
So, so binning is putting together all the genomes that belong together. So we group them to small groups, and a group ideally will represent a genome. Again, we use machine learning algorithms that we don't need to know exactly how they work. And apparently some people can do this manually. Maybe some geniuses, those who are patient enough. So what is done is that we look at the previous step of mapping to the genome, and we have the information of how the scaffolds are represented in different samples. And based on these metrics, you can have all the scaffolds which have identical metrics grouped into the same groups. For example, here, scaffold 1 and scaffold 2, they are almost identical in most of these metrics, so they belong to the same bin. So these are the correlation coefficients where you see that scaffold 1 and scaffold 1, they have a correlation coefficient of 1, which means they are very correlated. But scaffold 2 and scaffold 3, 2 and 3 will have a very low correlation coefficient, which means they're not binned together. So again, we have a lot of tools that we can use. Some of these tools are graphical user interface. Some of those are web-based, others are command line. So depending on your level of familiarity with one or the other methods, you can pick which one is best for you. And there's a lot of tutorials on some of these that I will show you tomorrow. So now the binning versus marker-gene, these are the differences between these two, and when you would want to use one versus the other. But the binning, most important is because it may include biases due to genome size differences and also horizontal gene transfer. So functional analysis, it works the same as the taxonomic analysis. What changes is the databases. In the previous taxonomic analysis, you use a database of taxons. Here we use a databases of functions. Functions can be things like antimicrobial resistance, glycolysis, and also gene families like beta-lactamase. Sometimes you cannot go directly to a gene, but you can know, okay, so this genome, we don't know which gene it is, but we know it's a beta-lactamase. So what you want to have at the end of your functional analysis is to group your some genes, your function according to the taxons. So for example, we know that if we have this number of genes, we know that more of these genes are coming from this genus 2 as opposed to genus 2. And then we have those differences in different samples. So we know that this sample, for example, has a higher functional ability than that same sample this gene in another sample. So these are different databases. We'll just look at them tomorrow, a couple of them. All those databases have pros and cons. Some of them are more appropriate for microbes like Metapsych, and then others are just specific like Resfine and CARD, which are just specific for antimicrobial resistance. These are, again, their availability on the web and how these are, whether they are web-based or graphical user interface. So some of these will require, of course, an advanced bioinformatics skills, because these tools that we have here will come with custom databases. But some of the databases we want to use, for example, in this paper, they want to look at the functional antimicrobial resistance using ResFinder. ResFinder will not be built into those tools, so we'll have to do custom analysis to align our reads to this antimicrobial resistance database, which may require more advanced skills, which are easily accessible if you give it some patience and read tutorials. So this is my last slide. Again, so at the beginning, we talked about the advantages of shotgun metagenomics, including the avoiding primer bias, and also being able to identify all the microbes and all the organisms, including eukaryotes, and also doing a direct functional profiling instead of just prediction. But of course, they will be more expensive and may not be able to get to the rare microbes. One of the things I want to talk about is that just because you found a gene does not mean that the gene is functional. So it is, again, that's why we're talking about, instead of saying we are determining the functions, we are rather determining the functional potential, because we find genes, but it doesn't mean that those genes are active. That's where another area of metagenomics that we call metatranscriptomics comes in, because this will rather measure the community activity by looking at the RNA, the messenger RNA, instead of looking at the gene itself. This is out of the scope of this workshop. And I thank you for your kind attention. It was difficult to fit it in 1 hour, but tomorrow we'll hopefully understand more about some of these principles we talked about today.
Thank you very much, Morris. That was a great talk. I learned too in the process. There were certain things that there was gaps in my knowledge, and thanks for filling in that for me. Much appreciated. So what we'll do now is open up for questions for now, if that's okay. Dr. Zende, Dr. Wasker, if that's okay with you, let's open up and have a question answer session possibly. Either you can talk in or you can put in your questions to the chat, and one of us will read it and ask Morris to kind of go through it for us.
Yeah, Bhushan, surely we can do that. If
Great.
anybody is having any questions, they can directly ask, or please text in the chat box, please.
Yeah, thank you. Thanks, Wasker.
Yeah, sure, sir.
पुलिंग ठीक होता है क्या? एक ही लगेगा, ठीक है ना? ठीक है तो चलो.
Or Bhushan, what I can suggest, can we have the session at the end, after the presentation of Meera? I think that will be fine.
Yeah, let's do that. Yeah. Okay. Yeah. With that, let's go on to our second speaker. Our second speaker is Dr. Meera Sundaran Nair, and Meera is currently the youngest faculty member at the Penn State Animal Diagnostic Lab. She's an assistant clinical professor, and now she's working towards her board certifications in microbiology. And I'm sure she will ace it at her very first shot. I'm very confident she'll do well. So Meera graduated from Kerala Veterinary College, or Kerala Veterinary and Animal Sciences University in 2012. She then moved on to do her master's in 2016 in the animal science program at UConn under my very good friend, Dr. Kumar Venkatarayanan. And then she moved on to do her PhD with him, again, at the PhD in Animal Sciences at the University of Connecticut. And her major focus was food safety. So she was really looking at the molecular aspects of addressing foodborne pathogens as it relates to food safety. She then moved on to University of Minnesota to start her postdoctoral training. She finished a postdoctoral fellowship. And then in 2019, we were very fortunate that Meera sent in her application, and we were so excited. And she was one of the most eminently qualified assistant professors that we could hire. So we're really thrilled to have Meera. And Meera is now working on several projects. She sleeps very little with a young baby at home. So Meera is working on COVID. She's working on several projects on COVID. She's actually fully immersed in genomics and bioinformatics as a part of her project as of now. And hopefully she will start her own research projects in a month or two as things progress. With that, I will leave Meera to give her presentation. I had a chance to look at it. I'm really excited to hear about it. So Meera, take it away.
Thank you so much, Dr. Bhushan, for the wonderful words. Thank you. And I also want to thank each and every one of you to take this time to attend the meeting. I know it's almost dinner time there. I will try to make it a little easier to grasp it sooner. With that, thanks again. And if you have any questions in between, or if I go fast, just put a message in the chat. I will try to slow it down and then to explain a bit more in between if you want to, or we'll have the question answer section at the end. With that, let's begin.
Able to share your screen, Meera?
Yep.
Yeah, there it is.
Okay, that's good. You're seeing my slideshow, correct?
Yes.
Yeah,
Okay.
now it is visible.
Wonderful. So yeah, you can see here, this is our Penn State mascot, our Nittany lion. So I'm here in the Pennsylvania State University. As Dr. Bhushan mentioned, I just joined in October 2009 as an assistant clinical professor here. I'm trained as a veterinarian, and I have years of training in food safety. Now, currently at Penn State, I'm involved in veterinary diagnostics, pursuing some animals health-related projects as well as some translational projects. My research focus is mainly anchored in microbial ecology with the focus on animal health and food safety, which will come under the whole big umbrella of One Health. So today, the learning objective of this session is mainly to understand the concept of what is a targeted Amplicon sequencing, and comprehend some of the workflows involved in profiling of a microbial ecology. We will also try to evaluate what are the expected outcomes we can get from a microbiome-based study. So let's start with defining what a microbial community is. As you all may know, it's a group of microorganisms that share a common living space and in which they can interact and function in many different ways, can independently function or cooperatively function. So when we look in a shared environment like that, there could be a number of species. It can be 1, 2, 3, or hundreds. So it's actually a community of microbes in an environment which consists of bacteria, archaea, viruses, fungi, or even protozoa. So when we think about a microbial community, this is some kind of picture which comes to our mind, right? So what do we learn from a microbiome study? Yes, we will come to know who is there, actually what's a representation of different species of microbes, what the abundance of species, or what they are doing there, what functions these microbes do, what are the metabolites they consume or excrete. Also, we can learn about how they interact with different hosts, right? Human host or animal host. That's some other piece of information we get from a microbiome study. This slide is actually a little busy, I understand. This is kind of an overview of demonstrating how diverse the sample types can be, along with what are the commonly utilized sequencing platforms and like systematic and stepwise processing steps. Today, with the interest of time, I'm only going to talk about the Amplicon sequencing and single gene target approach. And we heard a very good talk from Dr. Morris on shotgun metagenomic sequencing. So as he mentioned, metagenomics is a whole umbrella in which we can do different methods to understand the microbial community within it. It can be a shotgun metagenomic approach or a single gene target. So today I'm going to give a brief overview and concepts and approaches we take on the direction of single gene target approach, which is Amplicon sequencing. So just as I mentioned, the two most commonly used methodologies for microbial identification is based on either Amplicon sequencing or shotgun metagenomics. Over the past 25 years, gene Amplicon sequencing has been kind of a primary technique to study the phylogeny and taxonomy of several complex microbiomes. So in the case of Amplicon sequencing, what is important is we are targeting a marker gene other than looking at the whole spectra of genes, as Dr. Morris just mentioned in the shotgun metagenomics approach. In the case of bacterial identification, the most commonly used target gene is 16S rRNA, which is kind of a gold standard in microbial typing. This gene actually encodes the prokaryotic small 30 subunit of the 70S ribosomal complex in the most bacteria and archaea. And the question is why we should use 16S, right, for a marker gene identification. Yes, this is because all bacteria have 16S gene. It's highly conserved because it is necessary for the ribosomes to translate mRNA. And this gene has changed very little over time so that we can construct phylogenies to examine the evolution of the bacteria and their relationships to one another. This conserved nature also allows us to construct universal primers that can amplify sequences across many different bacterial groups. So that's why 16S have been used as a target gene Amplicon sequencing platform for bacterial identification. This is a 16S gene, which you see on the slide. It's actually of size around 1,500 base pairs in size. This gene actually consists of highly conserved primer binding regions. You can see these conserved regions are the green ones, which you see here, and the variable regions are the gray ones, which actually vary within the species itself. So what we can do is we can anchor our primers in these conserved regions and can sequence that region to understand about the evolution and the changes happened in different bacterial species. So for example, the most commonly used, the target is the V4 region. And you can see here to sequence the 16S V4 region, which is approximately of 254 base pairs, we can design primers surrounding these conserved regions. And then a MiSeq sequencing approach of, like, let's say, paired 150 base pair reads can actually sequence the whole length of the V4 with an overlapping region of approximately 46 base pairs. And it's also convenient that these primers get tailed with sequences to incorporate some adapters so we can do some pooling of the sequences to do it. So what happens after the sequences are generated? You will see different fragments of V4 regions, and we can actually put that into a reference database and see what one sequence, from when the sequence comes from, whether it's from E. coli, Bacillus, or Mycoplasma, depends upon the fingerprint of the sequences. This is the broad overview of the 16S Amplicon sequencing. Now, when we consider about a microbiome study or an Amplicon sequencing experiment, there are a few tips and tricks which will help us to better design the study. In general, any hypothesis should be primarily supported by a meticulous literature. It should be driven by evidence. Also, we can conduct some preliminary studies to avoid any uncertainties. So everything starts with the question you are addressing. You should have a specific question in mind. That's very important. Then think about the number of samples you need to have in the study. More the merrier, right? So selecting a significant sample size remains a key step. There are several literature and studies which have published looking at how to come up with a sample size estimation or the power analysis for microbiome studies. These are the two studies which have been cited in regards with the hypothesis testing and the power calculations for microbiome data. They actually describes different R packages like micropower and HNP to use to understand what's the best sample size to be used in different microbiome studies. The next important thing to consider is controls. We really need to have controls included in any study design. Otherwise, the signal which we are getting, we need to make sure it's kind of real, and not a spurious result. And the other important tip is metadata. We need to collect all appropriate metadata for each sample. That will give us the inferences which we are looking for, or our hypothesis needs to be answered, linking back to the metadata collected. Sample collection and handling requires much mention. There are this is a significant confounding factor that can affect results and interpretations. For example, see this image. This is actually from a study where they collected the samples from a teat canal of cows. They used two different methods to collect these samples. One is a dry cytobrush, the other is a wet cytobrush. It's kind of dry and wet sample collection method. You can see the differences in the abundance of bacterial species. Each color indicates a species. The dry ones had less number of species compared to that one of the wet sampling. So it's always important to consider when you design an experiment, what is your hypothesis? The question is very, very important. And based on that, you should describe your sample collection process. Nucleic acid extraction is another important tip to be considered, because many studies shows that DNA extraction protocols can cause differences in 16S RNA amplicon sequencing efficiency. For example, you see in this figure, the x-axis shows the different extraction methods. In one of the study, the others followed, and they are the y-axis shows the copies of the bacterial 16S rRNA gene per microliter. And they reported that the power soil kit was the one which gave the least number of bacterial copies compared to others. There was no significant difference, but there was some difference in the number of copies from these power soil kits. So that's something you need to consider too. What is your sample? How you are going to collect the sample, and how you are going to extract the whole genome from it. It's also important to consider which part of the 16S you want to sequence. Mostly people use V4, but that doesn't mean that you have to use V4 for your sequencing. You can use V2, V3, we can use V1. In fact, now different companies like QIAGEN has a screening panel where you can use multiple primer pools to figure out on your preliminary study which variable region works best with your question, hypothesis, and samples. Something like that can be made use of before you do your bigger studies on microbiome analysis. So again, this is a schematic overview of the 16S amplicon sequencing. So you start from the sample collection, you extract the DNA and amplify the region of interest, let's say V4, and we pull this and we do sequencing and we get the data out of it. So the data is like the ones which Dr. Morris mentioned, like FASTQ data, sequences which need some quality control, trimming and analysis. Those details I'll go over it tomorrow. As Dr. Morris mentioned before, there are a lot to talk about microbiome analysis and shotgun genomics. Combining everything in 1 hour is we are trying to do it. But today I'm not going to go over any tools, but we are going to get an overview and concepts and approaches which we follow in microbiome-based studies. So once we get the sequence out of it, there are 3 phases of processing. One is the pre-processing of reads, as you see in the phase 1, which includes the quality control analysis, trimming, paired and stitching, and even chimera detection. All these different steps we will discuss in detail tomorrow. The second phase is Otu picking. What is an Otu? Otu is the operational taxonomic unit. This is the reference or representative sequence we gather from the sequences generated, and the Otus are picked based on the 97% threshold similarity. I will explain a bit more about Otu picking in the next few slides. We pick the Otus, we align them, we do some phylogenetic tree creations, classifications, and look at the diversity analysis. That's an overview of the 60-days analysis workflow. So as I mentioned before, Otu picking or operational taxonomic unit picking is very important or very a critical step in the microbiome analysis. So what we start with is an experimental sequences, which is shown in the green. And we have a reference database like what you see in the blue box. So we align these experimental sequences to the reference sequences and see how many sequences that hit a reference. And the sequences with a 97% similarity threshold are grouped together, and we pick one of the reference sequences and we label it as the OTUs on different, like OTUs 1, 2, 3, 4, 5, 6, etc. Those ones which do not hit at the reference sequences will be done a de novo assembly, like they assemble based on the sequences and then go back and look for any undescribed organisms in the data set. So this kind of approach is called open reference Otu picking. This is most commonly used in many of the tools which you use for microbiome analysis. So just to explain it a little bit more simpler, so you have a lot of sequences which you are seeing as teddy bears there. So what happens in Otu clustering is those which look similar cluster around. And from these each clusters, which has a 97% similarity, one of this representative sample then goes back and align to the reference and see whether it correlates with E. coli, whether it's associated with Salmonella, whether it's associated with Klebsiella, and so on and so on. That's how the Otu clustering or Otu picking is happening. So it's kind of representative of what we have in the sequences. But definitely we get the number of Otus, meaning what are the different type of species and the number of species inside the sample which we are looking at. So again, recently in last 5-6 years, there is a new set of analysis which is getting more popular, which is called ASV prediction or Amplicon sequence variant prediction. How it is different from Otu picking is that in the Otu picking, what we do is we have sample sequences, we have Amplicon reads, we group them at 97% threshold and pick the Otu, right? But in the case of Amplicon sequence variant prediction, it's just simple. We are not going to group them into a cluster or like within a threshold. Rather, we take each sequences and align that to the reference. So we have a sequence variant rather than a collection of sequences. So what is the advantage of this Amplicon sequence variant is we will have less errors because anything which is as a sequence, a different sequence appearing in the data set, we take it and put it directly to the reference database and see what it's aligned to, whether it's aligned to an E. coli sequence or a Salmonella sequence and so on. So these are the two different kinds of approaches which we currently follow in microbiome analysis. Now more and more people are interested to do ASV prediction rather than OTU because of overwhelming responses on how this chimeras and PCR biases affects Otu picking. So this is something you need to consider when you try to start analyzing your microbiome data. whether it's an OTU or ASV, the taxonomy identity is always assigned to each representative sequence mapping to the reference databases. We have reference databases like Silva or Green Genes databases. These have been updated every 6 months or 1 year just to make sure that all the information is current and we have a good array of sequences there so that we can predict the presence of any microorganisms which we are seeing in diverse sample types. So once we have the sequences, OTUs or ASVs, and we have identified the taxons of different species, then we want to see what are the patterns in this microbiome. This includes the diversity analysis. So the diversity analysis has two ways of considerations. One is we can look at how diverse the how diverse the species within the samples, and we can also look at how diverse the samples are itself. For example, in the case of alpha diversity, we are in fact looking at what's the diversity of the species within the samples. So this is actually described with 3 different indices. One is species richness, meaning we can look at how many number of species or how many number of OTUs or ASVs we are seeing within this data set. It weights for all species equally independent of their relative abundance. That's what something like species richness gives out. We can also look at how abundant each species is. That's what it's denoted by evenness. When we say diversity, actually we look at both richness and evenness. So we are looking at number of evenly distributed species. I will explain a little bit more some pictures for your ease of understanding. For example, let's look at this. Community 1 and 2 of trees, right? In the first community, let's say we have 100 trees, 25 belonging to each of the 4 species. In the second community, let's say we have again 100 trees, but 80 of them are all one species. So what does that mean in terms of diversity and richness? This means that these 2 communities have 4 species. So the species richness is 4, irrespective of that, irrespective of the fact that how abundant they are. But when it comes to diversity, since the community 1 is more evenly distributed, it has a greater species diversity. That's what actually the main differences between species richness and diversity. That's what we meant by alpha diversity of a microbiome dataset. Little bit more for clarity, we can also look at how the species richness and diversity changes between different communities. Let's look at community A. This is kind of a perfectly even distribution. And the species richness, let's say it has 12 species in it. So the species richness is 12, with a Shannon and Simpson are in diversity indexes. I will talk a little bit more about it in next few minutes. But you can see here in a perfectly even situation, from to a highly uneven community, the Shannon indexes are decreasing. That means as the community gets uneven, our diversity decreases, whereas the species richness remains same because it doesn't depend upon the relative abundance of the species. So this picture clearly defines how species richness remains the same while the diversity increases or decreases based on the distribution of the species. And for alpha diversity, to describe the alpha diversity in better terms, we have come up with some mathematical formulas to calculate what is a diversity is. For example, the most commonly used index is Shannon index, which takes into account of species richness, relative abundance, and we take a natural logarithm of their relationship. So what happens with the Shannon index is the number increases with richness and evenness, and Shannon index puts more weight on the richness than evenness. So it ranges between 1.5 and 3.5. Whereas there is another index called Simpson index, which also take into account of the same species richness and abundance, but it is more influenced by evenness than richness. So what I want to tell in this from the slide as a take-home message is, when you define the diversity of a microbiome or a microbial environment, it's better to define with different indices so that you can understand exactly what is going on. With Shannon index, you can understand more from a richness perspective, but with Simpson index, you will get more of an evenness perspective. So it's always good to have different indices described for one data set, which will be easier to interpret your hypothesis or the rationale. So this is one of the studies published in which they looked at alpha diversity in cattle fecal samples from an E. coli 0157 super shredder and non-shredder steers. What I want to show you here is, you can see how this richness and Shannon indices differed among these groups. Between the non-shredders and the super shredder group, the richness actually was higher in the shredder group. You also see similar kind of reflection in the Shannon index, meaning that as the richness increases, the diversity also increases here. That means the richness and evenness, the abundance of the species also increases in the shredder groups compared to the non-shredder. This is the way you have to describe the alpha diversity when we analyze the data set. So before I jump into beta diversities and other diversity indices, I also want to mention that this kind of alpha diversity indices are prone for bias. For example, if we have different sequence length or different sequence reads, that can affect the interpretation of the species richness or number of OTUs or ASVs we see in the data. So it's always important to look at the rarefaction curves. What is a rarefaction curve? This is what this figure shows you. This is important to determine whether we have achieved enough sequencing depth in order to capture the full diversity within a sample. This is actually plotted with the reads and the sample size. So this is showing that an increase in number of species as the number of sequence reads increase. And at some point it gets flattened, meaning even if we sequence it further, it doesn't going to change the diversity of the sample. So it's always important to look at the sequencing depth and the diversity of the sample by doing the rarefaction analysis before doing the alpha diversity estimation. It gives us an idea that we are not biasing it to any side, and we have a normalized data set to do this. So this other approach of analyzing the microbiome data is looking at the beta diversity. As I mentioned before, alpha diversity, when it looks within the samples, beta diversity actually looks between the samples. What is the diversity? What is the richness or abundance of species between the samples? This is a measure of differential diversity between the ecosystems, and it takes into consideration of presence or absence or abundance of taxa. Let's look at this figure. Like, we have 3 mice. Let's say mice A, B, and C. When we look at the beta diversity, what we see is mice A versus B is 8 OTUs. And there is a difference of B versus C is 4 OTUs. And A versus C is 10 OTUs. That meaning this is what we are looking at between the samples or beta diversity. So we're looking at between A, B, and C, what's the difference in the number of species or the diversity we are looking at. Similar to the alpha diversity, for the beta diversity also, we have some indices or measures that broadly based on 2 different approaches. One is a sequence-based approach. When I say sequence-based approach, it can be qualitative or quantitative. In a quantitative sequence-based approach, what we are looking at is the sequence abundance. that was defined using Bray-Curtis algorithms or formulas. So Bray-Curtis approach actually looks at the sequence abundance, and it's more quantitative to define beta diversity. Whereas in the case of Jaccard indices, that's a different kind of index, which is a qualitative, which only considers the presence or absence of sequences. These are just names, like these are all backed up by mathematical equations and formulas. I'm not going deep into all those formulas and algorithms. Just want to give you an idea of when you define the beta diversity, they can be sequence based on the sequence abundance or the presence or absence of sequences. It can also be based on phylogeny based. Sometimes the phylogeny based outperform the sequence based. Why? Because that gives a clear idea of relationship between the sequence. For example, let's look at this picture. We have three communities. Community 1 with OTU 1, 2, 3, community 2 with OTU 4, 5, 6, and 7, 8, and 9 on community 3. When we just look at these three different sample sets, if we are just looking based on sequences, they all are clustered differently. You see three different circles in a graph down there. That means they all are different and they all have different OTUs and they are not even related. But when you look at phylogeny-based kind of diversity measure, all on the top are mammals and the last one are birds. So they group together and we have a one cluster of circle which is apart from the other two. So that's how the phylogeny-based diversity measures outperform. So you need to define what you want to answer from looking at these samples. It's always important to do these analysis on two different approaches, so you are not biased on any of these sequence-based or phylogeny-based strategies. That's something you need to consider when you analyze your microbiome data set. So with these numbers, which we derived using this abundance and sequence abundance or presence or absence of sequences, and with phylogenetic relationship, we build a matrix. And we will have numbers for each samples denoting the distances from one another samples. That will give you a distance matrix. So this distance matrix are then we can visualize using ordination methods. So when I say ordination methods, what does that actually meaning is, you can see on this figure here, we are trying to visualize the underlying structure variation of the data. So this is kind of a three-dimensional approach, but using this ordination methods, this multivariate data, we are trying to reduce the dimensionality and trying to put them in two different axes. And here you can see in this figure, we are looking at samples from different sites like gastrointestinal, oral, skin, nasal, urogenital. All these are clustered in by themselves based on the sites. This is the way the ordination methods work. We have a distance matrix created for all these samples, and we were able to see how they are grouped together using these scatter plots. We use commonly two ordination methods. One is called principal coordinate analysis method. The other one is non-metric multidimensional scaling method. How they are different is, ideally they are similar, but the way they rank these distance matrices is what that matters. For NMDS, they do ranks instead of just the numbers. For PCOA, it's more like Euclidean distances. So they both work on any desired distance matrices. You can use Bray-Curtis, you can use Jaccard or phylogeny based. The numbers can be easily plotted onto this one of these ordination methods, and we can clearly see what is going on with our sample. So that's what that's how we visualize beta diversity measures. Okay, so now we what we have done so far is we figured out what are the OTUs or ASVs or sequence variants in the data set. We aligned them to a reference database, and we understood what are the species or taxons involved in our samples. Then we looked at or samples, like whether the samples are diverse within itself or how the samples are diverse between the other samples or among other samples. So the one other thing we can do with this data set is we can also look at how differentially abundant the OTUs are. For example, I have a control mice which were we gave a treatment on the other set, and we can look at what are the different OTUs differentially abundant in these different treatment groups. So one of the most effective way of finding differentially abundant taxa is by characterizing the species indicator values, which means it combines the taxon's abundance within the sample along with the frequency of occurrence within the group. So if an indicator value is close to 1, then the taxon is highly abundant within the group compared with other groups, and it's present in most members of that group. For example, let's see this data set. It has two different set of data. One is an early fed mice group and the other one is a late fed mice group. And you can see which are the differentially abundant OTUs in early fed and late fed. This gives you an idea about what was going on in the microbiome of those mice which were early fed and late fed during the study. So this is kind of interesting to correlate, to give a causal relationship to what you're looking for. So marker identification or differentially abundant OTUs gives more inferences to your hypothesis or rationale what you're looking for. So as a summary, again, what we discussed so far is we looked at how we profile the data, how the alpha diversity is evaluated, how we can do the beta diversity evaluation. And as I mentioned now, like we can also look at the abundance of species by marker analysis. And tomorrow we will go through each of these steps using different tools. And I can explain with a data set how we visualize the differences among the groups and between the groups and so on. So this is kind of a bird's view overview of what we do with a microbiome data set. I'm hoping you got some concepts and approaches which you can adapt to your studies. So with this, I would like to tell you that 16S sequencing is a valuable resource. If you, it has its own limitations, and also it's important to remember that it's not the end of your question, but it will be the beginning of your question. So you can explore more relationship and network analysis and everything using this data set, and you can come up with different questions to be answered. So with this, I will open the floor for questions, and we will continue discussing the tools tomorrow.
Thank you, Meera. Wonderful presentation. Thoroughly enjoyed. Great set of information. With that, we have finished our presentations for today, and let's open up and see if there are any questions, and we can interact with the audience here. And we'll come back again tomorrow at the same time. We'll start at the Indian Standard Time around 6 p.m. And we'll start off with Morris, followed by Meera again tomorrow, in order to go and look at some of the practical applications of metagenomes and bioinformatics as they have stated in their presentations. So with that, let's talk about something that is more exciting for you. What's going on in Mumbai? What's going on in India? What can we do to work with you guys on genomics and bioinformatics?
Ma'am, I have a question. Yeah.
Can you please identify yourself first?
Yeah, I'm Dr. Meera Sundaran from Mumbai. My question to Dr. Meera is, you talk about frames of, reference sequences. there could be instances where you do not have a say you have a niche where you don't have a reference. In that case, how do you go about doing such an, how will you adapt this approach to such a situation?
Yeah.
Say you have a very unique kind of a sample which you got from a unique niche, for which properly there may not be a reference available. Or say a very unique disease state where you don't have a reference. Then how do we go about it?
Yeah, good question, Meera. So that's when we need to do de novo assembly. So it's a different approach than the reference set. We need to assemble it and blast it to see whether it kind of aligning to any of the sequences or it's there or not. We cannot use the reference data sets, and then we cannot use the open reference method too. We have to go for de novo assembly, like what we do for normal blast analysis and things. Did you get the answer?
Yes, thank you.
Mm-hmm.
I have one question.
Yeah, go ahead. Yeah, go ahead.
Yeah, this is Dr. Mudassir Ali. I'm from Scots, Kashmir, Jammu and Kashmir. I have a question to Dr. Meera.
Rather, it's a little difficult to hear you. Can you speak a bit louder, please?
Am I audible now?
It's not very clear. Yeah.
Maybe a network issue. Am I audible?
Meera, can you hear him?
Can you hear me, Dr. Meera?
Yeah.
Okay, it was quite an informative lecture to me. I'm actually naive to the topic, to be frank enough, but it was quite informative to me. Rather than question, I want to learn. That's why I pose this sort of a question to you. What I could get from the lecture is that if we want to know the microflora which may be present in a fecal sample or a milk sample, we need not to go for either isolation of the organisms. We can just use this technique and we will be able to identify different species that may be present in this a dung sample or a milk sample. Am I right?
Correct.
What my question is that it's based on the presence of DNA in that sample, which we will be extracting from the dung or the milk sample. What about the amount of DNA which we may require so that the amplification is proper and we will be able to identify the organisms properly? Sometimes the number of organisms in the milk or the dung sample may be too less to have enough amount of the DNA in the sample to extract enough amount of the DNA.
Okay, so your question is when we should start with the DNA, right?
No, my question is, do we require a minimum amount of DNA to be present in that sample so that we will be able to identify the organism?
Hello.
Am I audible?
Meera, this is Dr. Jagde. I think this Dr. Mudassir is asking about without isolating organisms from meat or milk or the fecal sample. Can you identify these organisms and how much of the load of the DNA is required to be present in that sample or item? So that he could be able to identify the organism. This question I understand. Am I right, Dr. Mudassir?
Yes, exactly, sir. Yes, exactly.
Yeah.
Okay, so yeah, definitely you need the amount of DNA in your sample. And it needs to meet some quality and quantity criteria to do the sequencing. And it's based on how you want to do. For example, Illumina, if you're using Illumina platforms and the libraries from them, they have a requirement where you start with. Dr. Bhushan, are you saying something?
If you are not speaking, can you please mute your microphone? Okay, let's let only the speaker unmute and then talk, because we are getting a lot of echo in the process. So go ahead, Meera. Yep. Meera?
Yeah.
Yeah.
So what I was saying is, Dr. Mudassir, like you need to start with a minimum level of DNA. The quality and quantity is it's critical, but you don't need a like each library kits which you are going to use to do the sequencing and downstream processes. They recommend a level of DNA to be there in your samples. So it can be anywhere from, studies have done looking at sequencing from 1 picogram through nanograms of quantities. So it depends upon how much reads you're looking for and what libraries you use. You need to have a minimum quality and quantity of DNA in your samples.
Okay, thank you. Thank you, ma'am.
Maurice, do you have anything to add to that, to your shotgun metagenomics?
Yeah, actually, so like you said, for 16S sequencing, where you have to do a PCR amplification, usually you don't need as much DNA as what you would need to do a shotgun sequencing, for example, because with the amplification, you make sure that you will be able to amplify even those with a rare with a low abundance. So some laboratories usually will require to send approximately 200 nanograms. So if you send 200 nanograms, now it will depend on the volume, how much volume you have. So if you have a decent amount of DNA, that would be a small amount of your volume. So yeah, 200 nanograms will be fine for 16S. 200 nanograms per sample, for each sample will be fine. But if you are working with a very special sample for which you can't have the 200 nanograms, still there is a possibility to still sequence with 16S, because you'll be able to amplify that. Just let you know that even if you send 200 nanograms, it's because of all the processes that they go through during library amplification. Usually the amount of the DNA you load into the sequence is really very, very low in the amount of only 5 nanograms, sometimes even in terms of picograms. But the starting DNA has to be enough, because most of it will be lost during the library preparation processes.
Thank you.
Ma'am, I have one more question. Can we use this kind of an approach to say identify either the unknown functional properties? I mean, it can be used as a tool to identify, say, some kind of functionalities which are not known till now, either by both approaches, whether it is Amplicon or shotgun.
So, Meera, for shotgun metagenomics is what we get more of a functional analysis, but we can definitely do some inferences from 16S microbiome studies using some downstream softwares like PyCrust and things like that. We can look at the functions of the microbes, which we see in the 16S. But while we pursue the shotgun metagenomics, as Dr. Morris mentioned, we can look at the functionalities of the group. So definitely we can look at the newer functions or any other undefined functions if you are expecting to see in a unique niche. Shotgun metagenomics is an exploratory tool to understand what's going on there. I will let Maurice to explain a little bit more on his part too.
Thank you. I think that is a good question. In fact, one of the uses of metagenomics is to explore, to be able to find new things. So because metagenomics does it does taxonomic analysis, but also, and most importantly, it does this functional analysis. So if you are interested in finding new things, if your target, your goal is to be able to potentially find new things, either new taxons, it could be that you want to find a new virus, it could be another new pathogen. You are interested in your ability to be able to find something new. In that case, that's where we go towards an assembly-based analysis instead of reads-based analysis. I think we talked about that, maybe not enough as we would like, but if you do an assembly, you'll be able to find something during the annotation. So you annotate a gene, you annotate something, you know it is a gene, but you can't find it in any of the databases. This is some of the things that people do to find new genes. See, it will be annotated as a hypothetical gene, but when you do further analysis like transcriptomics, you can find that the gene is correlated to a particular messenger RNA. You'll be able to say, okay, so there must be a function which is not annotated in the databases. So that's how they find new genes, new functions to gene. It can sometimes even be known genes, but which have been mutated, which have evolved over time to perform new functions. Yes, it's possible, and that's what metagenomics is there for, not only to find new taxons, new organisms, but also new functions. But of course, that will go further because the metagenomics will be an exploratory part of it, which will be followed by more analysis, more functional analysis.
Yeah, just to add to that, as we both said, this is a powerful tool with its own limitations, but to explore, this is actually a beginning to answer many questions which are unknown. So you can look deeper into the environment and see what microbes or what functions they do. You can explore newer and novel functions which are unknown too.
Thank you very much, both of you.
Maurice, could you explain especially one aspect of metagenome analysis that you're involved in? Is if I go and collect fecal samples from chicken or rectal swabs from chicken, and I'm trying to look at a metagenome or the microbiome profile, well, what is the first thing that's really you're going to be doing in terms of looking at the host DNA? How do you get rid of the host DNA in order to have a better appreciation of your microbiome that's there?
Yeah, I'm sure that's so. There are two ways to do that. So mostly people do sequence everything. So you collect a sample and then you do you remove. You don't remove anything. You sequence the full DNA, and then during the analysis, you use. That's what we will even do tomorrow. You do an alignment of your reads, your raw reads, to the genome that you want to get rid of. So for example, tomorrow we'll use some samples from pig farms where you suspect your host genome is going to be pig. Then you go into the NCBI or other databases, you find the genome, and then you align the reads. All the reads which will align to the genome, you remove them because you don't want them. That's one approach. Another approach that people do is to remove eukaryotic genomes before sequencing. People don't like that much because that process, you may also lose some of the microbial DNA.
Okay, good point. Yep. So it again comes back to the very question which Dr. Rather had earlier asked, is the amount of DNA required to throw into a machine. Is when you start doing it, if your microbiome or your microbial population is to begin with very small and your host DNA is pretty large, you tend to lose a lot of it in terms of noise. So I think where you calibrate your analysis becomes very important at that point in time.
Just to reiterate, the question is important, where you want to start from.
That's where I took your cue from your last slide, Meera.
And Dr. Bhushan, I think that's better, because that's exactly what we're doing with the project in the lab. This single primer
sequence independent CISPA analysis.
Yeah, the CISPA and GS. So because in this assumes that your sample is precious,
right,
and that you don't have enough. You can't afford to sequence everything because you may lose some of your DNA. So then you decide to amplify randomly, to amplify without targeting anything, just amplify anything you have there, especially when you have removed the host DNA. That will give you increase the resolution of the pathogens, or the detection limit. It will decrease the detection limit of your experiment. Yeah.
So the other aspect, which we will hopefully not be covering in tomorrow's presentation, and people who have the money with them, like Dr. Zenge, he's going to ask all of us, how much is it going to cost per sample?
Yeah.
So I know that's where the rubber meets the road, and we are all going to say he wants to do a project with 1,000 sequencing analysis done on a machine. How much is it going to cost me? 1 crore of rupees or 1 lakh of rupees? So what are your thoughts? What do you think is the cost of, so Meera and Morris, we have struggled with this issue for a long time. So if we can throw some light on this, the people who have the purse strings will have a better appreciation where we are going with this project. Go ahead, Morris. Yeah.
So I think at the beginning of my thread, when I was explaining the amount of data that you need to achieve this goal, if you want tomorrow, I have a formula that is used to calculate the amount of money you might need.
That would be great. Yeah. Yep.
So if you want tomorrow when I start doing, before I do the demonstration, I can show you, how people do that, and
okay, good. Yeah. Excellent. Yeah, that will be helpful.
Just an overview, those people who want to start a microbiome study or metagenomics, and it's kind of exploratory, as Dr. Earlier we got a question. If you want to just start with looking at the microbial population there, you can start with 16S. You don't need that much sequencing depth as a metagenomics. So this is cheaper too. So start with that. If you get meaningful information about the microbes present in the environment or niche you are looking for, you can always, you can start with 100 samples, and if you see some kind of signals there, I'm just saying an example, you can, the sample number can be reduced and see in smaller subset of samples the metagenomics to see the function wise or the network analysis wise what's going on as functionally to answer your question. So always you can start from the 16S and then go down deeper into shotgun genomics approaches to answer more and more questions. That's a way you can start with too.
Thank you, Meera. Any other questions? Meera, would you mind sharing your experience doing COVID-19 sequencing that we have done in our lab here?
Yes. So this been a crazy year for all of us, I know, and there's had been interesting year for us as researchers too, to looking at what's going on with this pathogen, evolution, mutation, variation, so and so. So here in Penn State, I have been involved in different SARS-CoV-2 studies, both a community surveillance aspect as well antibody like serology assay developments, genomic sequencing initiatives. And also some pathogenesis aspect too. So the genomic sequencing aspect, what we are currently doing is when we get some positive samples from the communities with the permissions to sequence, we sequence them and see where are we standing in a global epidemic, what strains, clades we are looking at, what's going on in the community. That's one aspect of genomic sequencing. So we have established a workflow to amplify this SARS-CoV-2 genome sequences from different type of samples, from cell cultures, from saliva, and so and wastewater even. So we have standardized different protocols to sequence SARS-CoV-2 genomes from these samples. So far what we have doing is like what I said, we look at the community samples and see what clades or what isolates are currently circulating. We also try to study mutational landscapes in different cell lines. We use these SARS-CoV-2 genomes to passage it to different cell lines. So we want to see, are they evolving or adapting in a very rapid phase? So we are trying to learn that with sequencing too. What are the mutations we are seeing in vitro? On the other side, we are also looking at wastewater genomes just to see what is circulating in the community on a different perspective, and are we seeing different variants popping up in the community before we know them from the people. So that's a different set of genomic surveillance we do. So we have 3 different approaches to understand the evolving issues with SARS-CoV-2. Yeah.
Thank you. Thanks. Any other questions for Meera and Morris? If not, great presentations, thoroughly enjoyed. Thank you so much for coming on and willing to share your time and efforts on this. Much appreciated, Meera and Morris. And of course, thanks to Dr. Zende and his group and his team for inviting us. Much appreciated. Thank you.
Please feel free to have questions tomorrow when we come up. We can discuss more on questions you have.
Yeah, tomorrow is going to be a little bit more intense because they will be walking through different tools and techniques. Yeah. So there will be a lot of questions, but that's what we are ready for.
Yeah. Yes, probably after going through today's presentations, we can hope more and more on queries and questions in tomorrow's session.
Yes.
Because it will be more of the practical one. Yeah, it was very nice to hear you, Morris and Meera, on these important aspects. Yeah, thanks a lot for this.
Thank you so much.
Thank you, sir. I take this opportunity to thank everyone, and especially the 2 speakers, Dr. Morris and Dr. Meera. You are very nicely explained this very difficult subject in very easy way and understandable to all the participants. I thank you all for joining with us today and enlightening about this shotgun metagenome sequencing and microbial community analysis by both of the speakers, very nicely narrated. And I would like to especially thank my teacher, my guru, Dr. Jairao sir, for moderating this today's training program. And I hope tomorrow also we are joining at the same time at 6 o'clock. In the
6 o'clock, yeah.
Indian time. So, yeah, 6 o'clock. And I also especially thank Dr. Vaskar, he is co-chairman of our this training program, and Dr. Savani is a rapporteur. So once again, at last, all the participants joining and interacting with all the speakers and clarifying their doubts
about whatever they are using this bioinformatics in their postgraduate and the PG research. So I once again thank from my behalf and my team, and also from the MAPSU team, our authorities, for assembling today for this third day training program. And tomorrow we start 6 o'clock. We start our next day's training program. Thank you so much.
Okay, thank you, Dr. Zende. Thank you, Dr. Waskar, and please give my regards to Dr. Paturkar and Dr.
Yes.
Kurkure. Hopefully, we'll try and see if they can join us tomorrow. Okay.
Yeah.
Sure.
Thank you. Yeah.
Yeah.
So
Bye.
much. Have a good night.
Good night.
Thank you.
Yeah.
Good night.
Bye. Good night.
Good night.
Good night.